So a very warm welcome to all of you who are joining us from across time zones and geographies to the fourth IUBS Centenary Webinar talk by Yvon Le Maho, and which is moderated by Renuka Badhe. Quickly, before we get started with the really exciting talk today, which is going to be on new technologies to investigate Arctic penguins without disturbance, all of us would like to know a little bit about who you are who are joining us in today's talk. It's always fun to get to know the audience. So we're going to have a quick audience poll. You'll see it pop up on your screens. Those of you who are joining us on Zoom, please do participate. Those of you who have joined us on YouTube or LinkedIn or Facebook, this should be shared. The link to this poll should be shared in the chat with all of you. So it's just two very simple questions. One is, where are you joining us from? And the second is, what are you currently doing? So we let this poll run for a few more for maybe like another 20 seconds, 30 seconds, and then we can move on to the talk. So I can see the numbers are kind of increasing. More and more of you are filling and participating in this poll. Okay, so maybe I'm ending the poll and sharing the results on the screen. So those of you who are joining us on Zoom, again, you should be able to see the results of the poll. You can see that a large percentage of you, 84% of you, are from Asia. We have some of you joining us, 3% from Africa, 3% from North America, and 9% from Europe. We do hope that some of you from South America and Australia are joining us on either YouTube, LinkedIn, or Facebook. So in terms of what you're doing, it's quite a mixed response. So we have 22% of you are bachelor students. That's wonderful. 19% of you are master's students. We have 19% PhD students. Some educators, 10% of you are educators, and we have some who are, researchers in academia, just who don't fit into any of the student categories. And so with that, I think we have captured who you are, who's joining us as today's audience. It's really nice to see the diversity, both in what you're doing and where you're joining us from. And so with that, I'm going to hand it over to Professor Ella Shashidhara, President of IUBS, to welcome you to today's webinar and tell you a little bit about why and how this webinar series came to be. Over to you, Shashi.
Thank you, Shantala. Greetings to all of you who have joined for this webinar, in whichever the platform on which you are using. I'm on behalf of India International Union of Biological Sciences. A hearty welcome to all of you. I know we are just passing through the current pandemic, almost at the end of it, hopefully. And although we lost several million people of modern civilization, I guess most of us and you all have sort of come out of it reasonably healthy and having not lost your beloved ones and friends and family. The reason I mention about the pandemic is not simply because last 3 years we went through this pandemic, is also rightly connected to IUBS. International Union of Biological Sciences was established in 1919, just the end of the previous pandemic, the Spanish flu. And that sort of, in addition to that, just after the First World War, people felt that internationalization of science, scientific cooperation across the societies is very important to address the real life problems and also to bring harmony, peace and harmony in the society. And IUBS since then has been working in that direction. Its focus has been in the area of holistic biology, which includes evolution, ecology, biodiversity, conservation. And obviously it will look at how human activities impacts biodiversity and the ecology and habitat degradation. And also in the modern days, we are all worried about the climate change, and IUBS is, focusing on many projects, international projects, bringing people on one platform to address the impact of climate change on the larger, globally on biodiversity and the ecosystem in general. And zoonotic disease is also an outcome of habitat degradation. And in that context, IUBS is focusing in modern days also to understand the One Health in holistic way, and see how we can actually save not only human life, also the domestic animals as well as wildlife by containing the spread of diseases. And that one of that it has to be to prevent the zoonotic diseases. In all of these things, most important aspect is democratization of knowledge and free exchange of information. And this webinar is in that direction. And we initiated this when IUBS turned 100 years. It's a centenary webinars. And this is the fourth webinar in this series. And thanks to Professor Yvon Le Maho for consenting to give this interesting talk, very important and timely talk on working on Antarctic penguins. There are two aspects to the penguins. One is, of course, penguin as a species and its biodiversity and its conservation is extremely important. And also they are the sentinel species to understand what's happening to the Antarctic ecosystem in general, because of the climate change. So studying penguins has two aspects to it. One is to conserve penguins as well at the same time to also understand the bigger picture about how climate change is impacting the globe. And Antarctic ice melting is one of the telltale signal of the impact of climate change. So in that context, again, it's very timely, and thanks to Yvon for consenting to give this webinar. I hand over to Renuka Badhe, who is the moderator. In all our webinar series, not only the speaker, even the moderator has always been an accomplished scientist in the field. Thanks to Renuka for agreeing to moderate this particular webinar. She herself is an oceanographer working in the area of polar research, particularly Antarctic research. And her specialization is phytoplankton physiology. But in addition, she's also specializes in economics and public policy, and in general, working in the area of the climate change, its impact, and how to mitigate the same. Renuka, over to you. Thank you very much.
Thank you very much, Shashi. I'm really happy to moderate this webinar. Yvon and I have been long-term colleagues, so I know of the work that he's going to present, and I'm super excited to introduce Yvon to you. If you could move the slide a little bit, please. Yeah, perfect. So Yvon has been an emeritus director of the CNRS, or the French Research Institute, based in the University of Strasbourg. He also is placed with the Monaco Scientific Center and is currently a member of the French Academy of Sciences. He has quite a few distinguished placements, like being a member of the Norwegian Academy of Science and Letters, and quite a few others as well. He's been one of the pioneers of using the gadgets like those that have the radio frequency identification placed with the penguins in order to investigate them without disturbing them too much. So without too much more introduction, I hand over to Yvon. Please go ahead. I will remind you about 5 minutes from the finishing of your talk. Okay. So please excuse me if I interrupt you at that time.
Thank you. Thank you. Hello, everybody. So I will share now my presentation. Bonjour de Paris. I'm in Paris now. Is it okay?
Yes, yeah.
It's okay. So my interest is on climate change and biodiversity. And then I will introduce you why I am interested in new technology to achieve my scientific goals. But first of all, I would like to make clear that climate change is only responsible for the loss of 7% of biodiversity, according to the Living Planet report of WWF. You see, in purple, it's only 7% from biodiversity, and habitat loss, for example, is about the double of this impact. And the most important factors are exploitation and habitat degradation change. The problem is that if biodiversity is reduced, therefore essentially due to human action, the impact of climate change will be larger. Solving climate crisis will not solve, obviously, biodiversity crisis. To act, to be efficient in our action, we therefore need to discriminate between the effects of climate change and direct human impact. But this is very difficult because of the superimposition of both impacts in most of our planet. A major interest of polar regions is that they can be considered as the only areas on Earth where direct human impact can be neglected. Of course, you have had a surexploitation of seals, for example, in the 19th century, but compared to the other parts of the planet, wildlife change in population can be used as a good indicator of the impact of climate change. And I remember that the first change were obtained of the clear illustration of the impact of climate change were obtained in the 70s in the west of the Antarctic Peninsula. It was at the Palmer Station that the biologist found that the population of Adelie penguins, here in purple, was decreasing, therefore starting in the early 70s. In contrast, the population of Gentoo penguins in brown sharply increased. What you have to know is that Adelie penguins are specialist feeders. They essentially rely on krill. And therefore, this drop in the population of Adelie penguins, already visible in the 70s, was due to a reduction, to a drop in krill in relation to the decrease in sea ice extent. In contrast, the increase in Gentoo penguins is due to the fact that Gentoo penguins rely on fish. And chinstrap penguins are about intermediary, which explain that they do not suffer from the drop in krill. What about other penguins, particularly in the subantarctic? We work in Crozet Archipelago, Kerguelen, in the subantarctic. Then it's much more difficult when you see a population like that, because if you see a change in the size of the population, is it due to another effect, to parasites, to some disease? Therefore, we had to find ways to monitor the change in the population, and the best way to determine what is the reason for a change in a population is to track individuals. And it's true not only for penguins, of course, it's true for many other animals. And I don't see why we should use only robotics or microelectronics to study the origin of life on other planets, like with Perseverance from the NADA, which was launched in 2020. And this is what I will illustrate in my talk. We had the fantastic development with microelectronics of biologging. Biologging is the use, as you see on the schema, with king penguin, which is equipped with a small instrument about the size of a coin. It's a Japanese instrument, which is monitoring acceleration, diving, which is giving the depths in relation to the time of diving and foraging. And therefore, it has been a revolution, because before that period, we were unable to know what was going on at sea, except if we were able to measure, for example, the amount of free fish or squid ingurgitated by these penguins. So it has been a tremendous revolution, the use of biologging. And today we have a lot of instruments, even on larger animals, which are able, like for example in elephant seals, to do oceanic studies using these instruments carried out by the elephant seals. And this is an example of what can be done on penguins, which is, it's a logger which provides information on acceleration, on depths, and according to what is registered, you are able, if you if you relate this kind of instrument with the behavior of the animal, you are able, when the animal is going back to his colony, to know exactly what it has done at sea. Satellite tracking has been also very useful to follow, to track penguins. And for example, in this data from my colleague Charlie Bost, we got information at the beginning of 2000, of the years 2000, on the trips of king penguins at sea. At the bottom, you have sea level according to the year. Here, this sea level indicates the temperature at the sea surface of the sea. And how it is obtained, it's obtained by a satellite, which JSON or Topex Poseidon, a satellite, which are 1,000 kilometers above us, and which are able to determine the distance between them and sea level with a very good accuracy, about one centimeter. And when sea level is higher than average, it's a warm sea. In the opposite, when the sea is cold, the sea level is below average sea level. Look now at the top of this picture, the data which were obtained in blue, the tracks of king penguins, and they show that when the sea is warm, the distance at sea is much larger. In the cold year, the birds are able to find their prey at what is called the polar front at a much shorter distance than when the sea is warm. You can see when it is about 500 kilometers or more than 600 kilometers. This is because the polar front, which is this where the subantarctic sea meets the polar sea, is much larger, and their favorite prey, which is myctophid fish, is located there. What is the consequence for the bird in the colony? This variability in the duration of foraging trips has a huge consequence. And if we consider, for example, the male, which is assuming usually in king penguin, the last period of the incubation, about three last weeks of the incubation, which means 3 last weeks fasting, while the female is at sea and will return at about the time of hatching. When we are in a cold year, the female has a short trip to do, and she will return before hatching. And she will then regurgitate food to feed the chick. And the male is leaving the colony. But what happens when we are in a warm year? In a warm year, the female usually comes back to the colony delayed after hatching. And we found that the male is able to store food. When arriving for this last shift, the male has food in his stomach, and this food is preserved. You see where we found it. And I will come back to that later, but it shows the impact of climate on the timing of the regurgitation of food, either by the female or the male, thanks to preserved food to ensure the survival of the chick. But does it have an effect? To do that, for many years, biologists would use flipper bands. And I remember then when I came at the beginning of the 70s for the first time in Antarctica, I was puzzled because I found that, and I was distressed, because I found that the so-called study sites were not full of birds as the control sites without disturbance. At that times, I was unable to study, to investigate why such a difference. I had to wait until almost 20 years with the discovery of the development of radio frequency identification, RFID, of which we were the first to use in penguins. It's a small capsule. It's less than 1 gram. You see here, 0.8 gram. We use the TIRIS system from Texas Instruments. And this small, very small, tiny capsule of less than 1 gram can be implanted under the skin, which therefore means that we don't have the when we started this study, the supposed impact of flipper bands. We knew at that time, thanks to my colleagues, I will show their name at the end of of this presentation, we already knew when we started with RFID that birds with 2 bands were delayed compared to birds with only 1 flipper band. And we also knew at that time that their breeding success was larger with only 1 band compared to 2 bands. But we ignored the effect of 1 band. And we studied that, coming back to this scheme, we installed underground antennas, radio frequency antennas, which allowed to monitor the passageway of penguins between the colony and the sea and back. With 2 antennas, we knew, according to which one was crossed first, we knew which direction the bird went. This is the only way we could do that, to track the birds at that time, because there is a very short reading distance. As I have shown before, the reading distance for radiofrequency tags is only 50 centimeters at most. So again, we had antennas installed on the entrance or exit of parts of the colonies which could only be entered or left using this 3 passage. And using that, we were able for the first time in 10 years later, about 10 years later, we were able to show that flipper bands induce, of course, I mean, it's clear for you, we had birds which were all of them equipped with radio frequency tags, and half of them were equipped with flipper bands. And we found that flipper bands introduced longer foraging trips at sea, a delay and a lower propensity in breeding, a 41% drop in their breeding success over 10 years, and a 16% drop in survival probability over 10 years. And this allowed us to show that there was a bias in using the breeding success and survival of flipper-banded king penguins as indicators of climate changes. But using radio frequency tags, obviously, we avoid the impact of flipper band, which is due, this was studied by Rory Wilson, a British colleague, which is due to the disturbance of when the birds disturbance at sea, the increase in drag when the birds are swimming. And thanks to the work of Céline Le Bohec, we were able to show that here it's in birds which were only tracked with radio frequency tags, we were able to show that a slight impact of the temperature at sea, only 0.3, has already a huge impact on survival. So this is using variability, normal variability, but of course, the predictions being much more than 0.3, you can deduce how important will be the impact of climate change on penguins, on king penguins. But radio frequency also allows to weigh penguins without disturbance. And we did that in Adélie Land, which is the south of Australia, we install a weighing platform, and using weighing platform, you see that it's possible to weigh penguins after and before and after their sojourn at sea. This one was very shy. It was his first passage, but now they are going back and forth, and therefore can be weighed. And this change in weight when they are coming back from the sea gives an indication of the impact of climate change on marine resources. But I wanted to do more. Here, it's an aerial view of one of the most important king penguin colony in Crozet, Possession Island. What I wanted to do is to know where the birds are located according to their experience. As I said, the reading distance for radio frequency tag is very short. And of course, it would not be possible to get in the center of the colony to study where those birds which are tagged are distributed according to their position as young breeders or older breeders, experienced breeders. So, and this is again, the difficulty is again because of the short reading distance. It would be very disturbing to go at the center of the colony. So therefore, the idea was to use robots, to use four-wheel rovers, which are walking into the colony. And we demonstrated that these rovers have no more effect. Here it's breeding king penguins equipped with a cardiac monitor, and using the heart rate as an indicator here, you see the huge difference between the effect of a human and the effect of a rover. In fact, as soon the increase in heart rate induced by the approach of the robot is about the same of a passing by penguin, no more. And in addition, when the robot is stopping, there is an immediate drop in heart rate, which shows that the bird is not disturbed. In fact, they don't bother at all as soon, even if the robot is very nearby, if not moving. In contrast with the human, there is still a huge increase in heart rate. And as shown on the left picture, as you can see, the presence, even this person is motionless, has a huge effect of disturbance because most of the birds which are around, which were breeding around, have left. And here, again, as you can see, if the rover is not moving, they don't bother. And if it tries, if it moves, they are fighting the rover as they would do with another penguin. For emperor penguins, it was more difficult because emperor penguins are huddling together. And as you know, they are huddling together during the winter. And would they have a territorial defense like king penguins? Obviously, they would not be able to huddle very tightly together. But the consequence, because they have no territorial defense, is that they are retreating on the rover, except if the rover is camouflaged, if the robot is camouflaged by a fake king penguin. And this fake penguin is thanks to Fred Olivier. Fred Olivier made a movie that you can see. It was produced by the BBC, by John Donner Productions for the BBC. And it's, I think, one of the few examples where the success of the study was done by a collaboration of filmmakers and scientists. And here, this is a camouflage chick in a huddle of emperor penguin chicks, which would allow the layer to do behavioral study with such a crash of emperor penguin chicks. Now, we want to do more. The problem with a small rover masked by a fake emperor penguin is that adult incubating or raising a chick are still very shy, in contrast to their behavior that you have seen with a camouflage, with a robot camouflage by a fake chick. And for example, here we developed a prototype of a fake adult that we wanted to use to study emperors during the winter. What we would like to do is to approach birds in huddles during the winter to know how much time they spend in the periphery into the huddle. And we develop this fake emperor penguin robot. However, although for us, this penguin seems fantastic, it was made by the industry of the costume, was made by the cinema industry. We had the help of very skilled people in this domain. It did not work. And presumably, it's because this costume did not look like it should for emperor penguins, which are able to see in the UV. And the UV image of this costume was presumably a disaster, according to that. So we are now working on a more design emperor penguin, fake emperor penguin, adult fake emperor penguin. It is now being built by engineers. So engineers are very important in these studies. In conclusion, new non-invasive techniques are essential to avoid human impact and the subsequent scientific bias when using wild animals as indicators of the impact of climate change. But non-invasive techniques are also the key when investigating the physiological adaptations of wild animals as a source of biomedical innovation. Another reason for the need to preserve biodiversity for future generations. You remember, I showed that before that a king penguin would be able to store food in their stomach. You remember this picture on the left, which made the cover of Nature. It was very fascinating that king penguins are able to preserve food in their stomach for 3-4 weeks. Fascinating because their body temperature is 37 degrees Celsius. So we studied the conservation and found one molecule on the right top, a peptide, which is a small peptide of 38 amino acids that is produced in the stomach and this is an antibiotic molecule. And we found that this antibiotic molecule is able, as shown at the bottom on the left, when added to Aspergillus fumigatus in a glass, you see here, or we have also an effect on Staphylococcus aureus. The spores of this fungus is disappearing with the penguin peptide. So now we just got a grant to study further this antimicrobial action of this peptide, which allows king penguin chick to survive when the female is delayed, to study, to fight what are the main factors of the so-called nosocomial disease in the hospital. But this would also not have been done without developing techniques to avoid disturbance, because to do that, we had to take stomach samples without disturbing the birds so that they would continue their incubation and their conservation of food in their stomach. Of course, these studies.
Yvon, just a quick reminder. I know you're very close to finishing, so
It's the last picture.
Perfect. Thank you.
The last picture. And of course, what I have shown would not have been possible without a larger team. And you see Fred Olivier, for example, which was a filmmaker, engineers, a mathematician, Victor, Charlie Bost, I gave you his name, Yann Robert Couder with biologists, Michel Gauthier-Claire was involved in with Yves Drich in food conservation, Céline Leboeck, which got all this information using the radiofrequency identification, and Claire Sarrault showed that in fact using flipper-banded birds do not allow to use penguins are indicators of climate change. And these three here, Philippe, Cécile, and Jules Hoffman, were very important for the discovery of this molecule. And Philippe Bullet is now conducting a new study to understand the mechanism of action of this molecule, which is very interesting because it has been evolved in marine birds. The stomach is saline, and we have very few molecules, antibiotic molecules, still efficient in a saline medium, particularly for ocular infections. And Jules Hoffman he got a few years after we worked together the Nobel Prize on insects. Merci beaucoup. Thank you very much for your attention.
Thank you very, very much. Wonderful talk, Yvon. To all our attendees, if you have any questions, please make sure that you use the Q&A box on Zoom and enter your question there. Or if you're watching on one of the streaming channels, either YouTube or elsewhere, please use the chat box at that function, and then we will pick up the questions and ask Yvon your questions. I think we have a few questions already with us. You know, you spoke about working with these penguins for very long. Can you give us maybe an idea of how bad the weather can get and how changeable it is when you're out working in the field with penguins? And maybe how bad it was the last time that you have visited Antarctica?
So,
Yvon, could you please stop sharing the screen?
Yes, I was able to do it. Yes.
Thank you.
So the last time I was there was I went 4 times for pioneering the robot study. Twice in the Subantarctic and twice in Antarctica. Last time was in 2013. And since then, I am developing new robots. The difficulty, as you have seen, is to be able to build a new fake adult emperor penguin because they are very shy. And although it worked very well with the fake cheeks, I don't know if you remember on the slide, they tried even to communicate with a fake cheek. Why it doesn't work so well? That only the unemployed birds, which are very curious, come close to the, we have to find that. So the second question was about
what kind of temperature ranges you have seen when you're working there.
Yes.
If you could describe some of the weather conditions, because I think a lot of our attendees of the webinar haven't been to the Antarctic, so it would be great if you could describe.
Okay, when I overwintered, because emperor penguins only breed during the winter, the lowest temperature we had were not so low compared to Canada, for example, or Siberia. The lowest in Dumont d'Urville were around minus 30, but it's one of the most windiest places on Earth. And we regularly had more than 150 kilometers of wind. And even the world record in Antarctica, I guess, once we had 330 kilometers an hour of wind. So I went almost every day in the emperor penguin colony, except one or twice, particularly when we had more than 300 kilometers, because it's very difficult to walk outside when the wind speed is over 150 kilometers an hour.
No, absolutely. And I think you may have covered some aspect of this question in your talk, but I think it would be good to respond again, especially since it gives a really nice segue to what you were talking about just now. Yes. Like you said, Antarctic conditions are very extreme. The weather there, even for the penguins, when they live there, they're used to these extreme weather conditions. So when we insert these RFID
tags
into the birds, do these tags get affected by the extreme weather? And how do you work to ensure that?
No, it's only done during the summer because obviously what we want to know is later on is the age of the birds. So we only implant tags on chicks, not on adults. We are not allowed to work on adults, and we are not allowed, which is good for the conservation, and there would be no interest because we want to know the age. And for example, in king penguins, more than 2,000 king penguin chicks have been implanted, which means 10,000, which means we are really working at a population scale.
No, absolutely. But what kind of failure rate do you get with these tags? I know you said you work in the summer, but from my experience, I know it can be quite horrendous weather in the summer as well. So what kind of failure rate do you get normally with these tags? Maybe
Okay.
not just due to the weather, but just in general.
Yes. So what is interesting to say is that therefore we only equip chicks, and to avoid implanting twice on chicks, we have to mark them with a number. And it's another important point. What I want to stress out is that when we started, we implanted a few adults to try first, and even 1 year after, they seem to be very anxious when you approach. So in contrast to chicks, and this is why we have decided not anymore to implant adults, because birds are too reactive. And now we are even working with, I'm doing that with Victor Planas. I showed this picture. Victor is a mathematician, an expert in machine learning, and we are combining machine learning and robotics, developing new techniques, for example, using individual recognition by imagery. So we are again developing, and we are also investigating something which is very exciting. The rover, the robot is an intruder, but why is it no more considered as a threat than another birds passing by in the colony? This is very exciting questions.
Yeah, absolutely. So this seems like super exciting work. Are there any other countries around the world that are doing something similar to this? Is this being caught up and kind of picked up around the world, I presume? That would be good to know as well, because it seems like a really good way to work with penguins and with animals in general.
Yes, I know that when we started at the beginning of the 90s, when we started radio frequency identification, the year after already, the Australians started. They already had before a system for automatic weighing, but without radio frequency identification. And the year after, they used it, of course, because it's very useful. Because again, if you have automatic identification, it allows you to have automatic weighing. And this is also what we are doing now in a zoo, because the veterinarians are very eager to be able to follow the health, the condition of the penguin, and the main indicator is body weight. The problem is that in zoo, usually they had to run after the penguins, and which is stressing, of course, and sometimes they got the information about their body weight too late. And now we have installed, for example, in Paris Zoo, we have installed a weighing platform. Automatic weighing platform. So it's not only for wild animals, but it's also of interest for captive animals.
So that's really good to hear. And there's a little bit more of a general question about penguins and global climate change. How do you see the population of the different penguins changing with the predicted 1.5 degree rise in global temperatures, with the COP, just going on right now on the side of what we're doing here?
Yeah, so again, the most important effect now is in the subantarctic, on the west of the Antarctic Peninsula. And for those who are attending this seminar, it's interesting to know, to quote, that this area on the west of the Antarctic Peninsula, with another area in Siberia, are those areas on our planet where the temperature change is the most important. It's interesting too. Considering the huge Antarctic continent, we have contrasted effect, for example, on Adelie penguins, because we have areas where the population are decreasing and other areas where the populations are increasing instead. But in the future, what will happen due to the decrease in sea ice extent is very, very intriguing, and we are very anxious about that because, as you know, krill is breeding according to sea ice extent during the winter. The sea ice has a huge importance for krill breeding, and krill is the main biomass source of food for many animals in this area, including the whales.
Cool. So we have krills, and we of course also have copepods that are important for different species of penguins. But I think it'll be, I know that there are some penguin species that like ice, and those are tending to get more affected by ice. And then just as an add-on to what you were saying, Yvonne, and then some species that don't like ice that much, they prefer to nest on open ground. And I think so this depends on how much effect
yes, and it's there is a compromise, in fact, because look, Adelie penguins, if the thick ice is too large, they have a distance much too large to walk until they find areas to feed. But if sea ice has disappeared, as I said before, again, there is less krill. So there should be a compromise.
Yes, apologies. The screens had shifted a little bit for me. I think that's that kind of global perspective of climate change and its effect on these amazing birds in the Antarctic. Thank you very much, Yvonne. I think this was a really nice, really nice talk. It was a pleasure listening to the talk and a pleasure talking to you after so many years. If you have any final thoughts, please feel free to share them with our audience. Otherwise, we'll close in about a minute's time.
My main message is for the planet, not only for Antarctica. It's that we will not save pollinators, for example, bees, by windmills, it seems to me that there is a confusion between climate change and biodiversity. And for those attending this seminar, I think the main message is that most of the change now is not due to climate change, most of the reduction in biodiversity, but it's very important because a fragilized biodiversity will respond very badly to climate change. If we want to adapt to climate change, we need to restore biodiversity first.
That's an interesting perspective. I might slightly disagree with you there, because climate change also directly affects a lot of species that are living in the cold regions, particularly. You also have something like the poleward shift of species that occurs as a direct response to climate change. So I think there's so many perspectives to be looked at when we talk about these issues. But yeah, like I said before, it's a fantastic thing to have a debate, to have a chat with you again. Thank you very much to all the audience that came and supported us and listened through to this talk. And yeah, thank you very much to IUBS for this centenary webinar. And who do I pass on to next? Do I pass it on to Shashi, or Shanta, would you take it on to finish off?
Shantala will take over.
Thank you very much.
Thank you very much, Renuka, and thank you for all of you.
Thank you very much, Yvonne. Thank you very much, Renuka. It was a really, really exciting and interesting talk. And for those of you who are still in the audience, if you're interested in the previous talks, you can visit the IUBS webpage, and there are recordings of all of the previous webinars and talks that have been part of this IUBS Centenary series. All of them are very interesting, exciting, and touch upon, like diverse and different aspects of biology. So with that, I think we can wrap it up. A very big thank you to all of you who joined us today across platforms. Thank you, Shashi, Natalie from IUBS, and the speakers, and the team from India Bioscience, who are the technical hosts for this event.
Merci.
Merci beaucoup.
Thank you all. Thanks, your wonderful talk. Really enjoyed.